Hi everyone, in Learning Outcome 2 of Module 2, we're going to be covering specifically the red blood cells, and these are a component of the formed elements. So let's get started. A single drop of blood contains millions of red blood cells, white blood cells, and platelets, and all of these are part of the formed elements of blood, and one of each type is shown here on this image. Like I said, the red blood cells are a component of the formed elements of the blood. It can also be called erythrocytes, and it's represented here by this cell. Then we have leukocytes, which are also the white blood cells, and these are going to be divided into granular or agranular. This just tells you that the ones that are granular will show granules inside of the cell, and the agranular will not show granules inside of the cell. And this image here is showing an example of a leukocyte. You can see that the size is similar to the red blood cells, but we will talk specifically about the difference in size when we get to talk about the leukocytes. And then we have the last component, which is one that's not cellular, and that's the platelet, which is represented here on this image by this structure full of cell extensions. So let's cover first in this learning outcome, more specifically, the red blood cells. Red blood cells, they can also be called erythrocytes, and they will account for slightly less than half of the total blood volume, which we can actually see from this image how it does represent about half of the total blood volume. In fact, when we see the hematocrit, which basically stands for the value that indicates the percent of whole blood contributed by formed elements, we see that both in normal adult males and females, they're close to 50%. For males, the average is 45%, with a range between 42 and 52%. And in females, the average is 42% with a range between 37 and 47%. So pretty close to half of the total volume. And because whole blood contains roughly 1,000 red blood cells for each white blood cell, the hematocrit closely approximates the volume of erythrocytes. And as a result of this, hematocrit values are often reported as the volume of packed red cells, or VPRC, or even simply they call it the packed cell volume, which stands for PCV. Now, the number of erythrocytes in the blood of a normal individual really staggers the imagination. Normal adult males will have 5.4 million erythrocytes per microliter, which is the same as cubic millimeter if it helps. And females will have about 4.8 million erythrocytes per microliter. And in addition, if you think about the amount of erythrocytes that are contained in one drop of blood, that would be 260 million. And the entire body of an average adult, that would be 25 trillion. Yes, trillion with a T. For that's amazing. I know amazing doesn't start with a T. I was looking for an adjective that started with a T. So we just add that's amazing for trillion. But it is very impressive. The erythrocytes, they will transport both oxygen and carbon dioxide within the bloodstream, and they are among the most specialized cells of the body. And their anatomical specializations, they're apparent when the red blood cells are going to be compared with your typical body cells. So each red blood cell is this biconcave disc, as we can see here on this image, that has a thin central region and a thicker outer region. And what is the importance of this shape? Well, this very unusual biconcave shape will provide strength and flexibility, and this will allow the cells to bend and flex with an apparent ease, and by changing their shape, individual blood cells, they will be able to squeeze through capillaries that are very small in diameter or that have been distorted or compressed. So by being flexible, the red blood cells will be able to pass through those capillaries. It also gives each red blood cell a very large surface area for a cell that's pretty small, because it's only on average about 7.8 micrometers in length and about 2.6 micrometers in thickness. And this relatively large surface area will allow a rapid diffusion between the red blood cell's cytoplasm and the surrounding plasma. And why is this important? Well, as the blood circulates from the capillaries to the lungs, to the capillaries of the peripheral tissues, and then back to the lungs, the respiratory gases are going to be absorbed and released by these red blood cells. And as they are the ones that will transport oxygen and carbon dioxide, by increasing the surface area, this exchange can occur faster. Also, this biconcave shape allows the red blood cells to form what we call stacks, like these dinner plates stacked on the shelf. These stacks are referred to as rouleaux, which means little rows, and they will form and dissociate repeatedly without affecting the cells that are around them or the cells that are involved in the stacking. And what is the importance of the stacking of erythrocytes? Well, one rouleau can pass along a blood vessel that is a little larger than the diameter of a single erythrocyte, whereas individual cells, they will bump on the walls, and they would also band together to form these log jams that could block the vessels. And it is the same as when you were, for example, in kindergarten, and right when it was time for you to leave your classroom and go to the bus stop, for example, or when you were leaving your classroom to go to the cafeteria and have lunch, your teacher made you walk in a line. And I bet you it was way more organized than trying to exit one of the large rooms at FIU with 300 students wanting to pass through a narrow door at the same time. It gets clogged, right? So now if all 300 students formed a line and walked one right after the other, the transition would be more orderly. So here is basically the same concept of the rouleau, to try to get as many red blood cells passing through the tiny capillaries. During their differentiation and maturation, red blood cells, they will lose most of their organelles. They basically just retain an extensive cytoskeleton, and as a result of this, the circulating red blood cells, they will lack, for example, mitochondria, endoplasmic reticulum, ribosomes, and nuclei. And where will they get their energy from without having mitochondria? Well, they can obtain energy only through anaerobic metabolism, and they also rely on glucose that's obtained from the surrounding plasma. And why is this mechanism so important? Why is it that red blood cells, they lack mitochondria? Wouldn't it just be easier to be a normal cell like all the others and have mitochondria? Well, this mechanism is important because it will ensure that the absorbed oxygen will be carried to the peripheral tissues and not sort of stolen by the mitochondria if they were present in the red blood cells. And the lack of ribosomes and nuclei means that the protein synthesis cannot occur. Therefore, a red blood cell is going to be unable to replace damaged enzymes or structural proteins. This is a big problem for these cells because, as you can imagine, they are exposed to very severe stresses, as a single circuit of the circulatory system usually takes less than 30 seconds. This means that we start counting once the cell leaves the heart and stop counting when the same cell arrives back to the heart. It only takes about 30 seconds. And in this time, a single red blood cell stacks in rouleau, contorts and squeezes through capillaries, and then joins their friends to rush back to the heart for another round. Now, with all this wear and tear and no repair mechanisms, A typical red blood cell has only a relatively short lifespan of about 120 days. In these 120 days, they will actually travel about 700 miles. Isn't this crazy? It's like going from South Florida to Tennessee. And in this daily activity, either the plasma lemma ruptures or the age cell is going to be detected and destroyed by phagocytic cells that are surrounding. This means that about 1% of the circulating erythrocytes are going to be replaced each day. And in the process, approximately 3 million new erythrocytes enter the circulation each second. Yes, each second. This is just crazy if you think about it. Let's say that this video will take maybe 10 minutes. At least this is my goal. In reality, it will probably be a little bit longer. But anyway, if it is 10 minutes long, this means that during the time that you will be watching this video recording, you will have produced 1,800,000,000 new erythrocytes. If this does not make your mind go crazy, I don't know what will. Well, like I said, a developing erythrocyte, they will lose all their intracellular components except for the ones that are associated with its primary function, which is the transport of oxygen and carbon dioxide. A mature red blood cell will consist of a plasma lemma surrounding a cytoplasm that will contain 66% of water and 33% of proteins. Now, the main protein is going to be hemoglobin that will represent 95% of the cell's protein. And functions of hemoglobin, well, is the protein that's going to be responsible for the cell's ability to actually transport oxygen and carbon dioxide. Hemoglobin will have a red color, which is what gives the erythrocytes its color, and the tones of red will change according to the amount of oxygen or carbon dioxide that it contains. Therefore, oxygenated hemoglobin has a bright red color, whereas deoxygenated hemoglobin will present as this deep red color. And this will actually account for the color difference between arteries and vein. Now, it does not mean that all arteries will be bright red because all arteries carry oxygen. When we get to the lecture about the heart, we will cover this in more details. Each hemoglobin molecule has a complex shape, as we can see here on this image. The molecule is going to be composed of 4 polypeptide subunits. There's going to be 2 alpha chains and 2 beta chains, and these are representing the globin part of the hemoglobin. And each of these globin subunits, they will contain a single molecule of heme. And each of these heme units, they will hold an iron ion in such a way that it can interact with an oxygen molecule. And this oxygen-iron interaction is very weak. And this is important because the two will be able to separate very easily without damaging either of the two, the hemoglobin or the oxygen molecule, when the oxygen is leaving the red blood cell and going into the interstitial fluid. Just as a curiosity, there are approximately 280 million molecules of hemoglobin in each normal red blood cell. And because a hemoglobin molecule contains 4 heme units, this means that each erythrocyte potentially can carry more than 1 billion molecules of oxygen. Let's not forget about the carbon dioxide. So in addition, hemoglobin also carries about 23% of the carbon dioxide that's transported in the blood. Carbon dioxide will bind to amino acids of the globin subunits rather than competing with the oxygen for binding with the iron. And the binding of carbon dioxide to globin subunit is just as reversible as the binding of oxygen to heme. And as red blood cells circulate through capillaries in the lungs, then oxygen enters and carbon dioxide leaves the plasma by diffusion. And as plasma oxygen levels climb, then oxygen diffuses into the red blood cells and binds to hemoglobin. And as plasma carbon dioxide levels fall, then hemoglobin releases carbon dioxide that diffuses into the plasma. Basically, in other words, the red blood cells, they will absorb oxygen and release carbon dioxide. Now, in the peripheral tissues, the conditions reverse because the active cells are consuming oxygen and producing carbon dioxide. And as blood flows through these tissues, then oxygen will diffuse out of the plasma and carbon dioxide diffuses in. So the red blood cells will then release oxygen and absorb carbon dioxide. Hemopoiesis is the process of blood cell formation, and the stem cells, which are called pluripotential stem cells or PPSC, are ultimately going to give rise to all the blood cells. And the process of erythrocyte formation or red blood cell formation is termed erythropoiesis, and they will start to be produced in the bone marrow. Therefore, a stem cell will differentiate into an erythroblast, which will then become a reticulocyte, and this reticulocyte will reach the blood vessels and mature into an erythrocyte. Now, how will this process be activated or controlled? Well, it is going to be controlled by a hormone that is going to be secreted by the kidneys, and this hormone is called erythropoietin. So this hormone will increase the rate of production of red blood cells in response to the falling levels of oxygen in the tissues. So once they are formed, they will live up to 120 days in circulation, and after which these worn out cells, they're going to be removed by the circulating macrophages, which are located primarily within the bone marrow, the liver, and the spleen. And then the components of the degraded erythrocytes, hemoglobin, are going to be further processed in the following way. So nothing is wasted, everything is going to be recycled. Globin, which as we know is the protein portion of the hemoglobin, it's going to be broken down into amino acids, which can be sent back to the bone marrow to be used in the production of new erythrocytes. And the hemoglobin that is not phagocytosed is going to be broken down in the circulation, releasing the alpha and beta chains that are going to be removed from the circulation by the kidneys. Next, we have the iron that is going to be contained within the heme portion of the hemoglobin, and this may be stored in the liver or the spleen, primarily in the form of ferritin, or it can also be carried through the bloodstream by transferrin to the red bone marrow for recycling into the new erythrocytes. And the non-iron portion of the heme is going to be degraded into the waste product of biliverdin, which is a green pigment. Verdin means green, and then it will be converted into another waste product, which is called bilirubin, and this one is now a yellow pigment. So bilirubin will bind to albumin, and it will travel in the blood to the liver, which uses it to then manufacture what we call bile, which is going to be a compound that's going to be released into the intestines to help with emulsification of the dietary fats that we consume. This means that if your dietary intake does not include fat, then bile will be stored in the gallbladder. But we will definitely discuss more about this when we get to the digestive system. For now, just remember that biliverdin is going to be converted into bilirubin, and then bilirubin will bind to albumin, which will then travel to the liver, where you're going to have the formation of bile that will be stored in the gallbladder for use with fat digestion. Here we have an example of a malformation of red blood cells. in this disease that's called sickle cell anemia, and it is caused by a mutation in one of the hemoglobin genes. So this will cause the erythrocytes to produce an abnormal type of hemoglobin, which causes the cell to take on a sickle or a crescent shape like this one showed over here. So sickle cells will block the blood flow to organs and deprive the affected organs of blood and oxygen. In addition, in sickle cell anemia, the blood is also chronically low in oxygen, and this lack of oxygen-rich blood can damage nerves, as you can imagine, and organs, including, for example, the kidneys, the liver, and the spleen, and can be fatal. Now, individuals that have this disease, they can lower their chances of difficulties from the disease and actually enjoy many normal activities just by getting regular checkups with their doctors, and also by preventing infections by taking simple steps, including, for example, frequent hand washing. So it's as simple as that, but of course, it can be very complicated. And an average lifespan is around 50 years old.